Dear students, happy to meet you in this online mode of education. I am Dr.S.ANGELINA, GLORITA PARIMALA, Associate Professor and Head from the PG and Research Department of Zoology, A.D.M. College for Women, NAGAPATTINam. The topic I am going to discuss with you today is about the Mendal'S law of inheritance, which is coming under the paper genetics. Let us see one by one. This law of inheritance is first promoted by Mendel. He is called as father of genetics. Johann Gregor Mendel, during the year 1822 and also till 1884, he is a father of genetics. He was a lifelong learner, teacher, scientist, and a man of faith. He taught physics, botany, and natural science courses at the secondary and university level. In 1856, he began a decade-long research pursuit involving inheritance patterns in honeybees and plants, ultimately setting on pea plants as his primary model system, that is, a system with convenient characteristics that is used to study a specific biological phenomenon to gain understanding to be applied to other systems. In 1865, Mendel presented the results of his experiments with nearly 30,000 pea plants to the local Natural History Society. He demonstrated that traits are transmitted faithfully from parents to offspring in specific patterns. In 1866, he published his book, Experiments in Plant Hybridization, in the Proceedings of the Natural History Society of Brünn. Mendel's work went virtually unnoticed by the scientific community, which incorrectly believed that the process of inheritance involved a blending of parental traits that produced an intermediate physical appearance in offspring. This hypothetical process appeared to be correct because of what we know now as continuous variation. Continuous variation means what? It is the range of small differences we see among individuals in the characteristics like human height. It does appear that offspring are a blend of their parents' traits when we look at characteristics that exhibit continuous variation. Mendel worked instead with the traits that show discontinuous variation. When we see the discontinuous variation, it is the variation seen among individuals when each individual shows one or two or a very few easily distinguishable traits such as violet or white flowers. Mendel's choice of these kinds of traits allowed him to see experimentally that the traits were not blended in the offspring as would have been expected at a time, but that they were inherited as distinct traits. In 1868, Mendel became abbot of the monastery and exchanged his scientific pursuits of his pastoral duties. He was not recognized for his extraordinary scientific contributions during his lifetime. In fact, it was not until 1900 that his work was rediscovered and reproduced and revitalized by scientists on the brink of discovering the chromosomal basis of heredity. When we see the Mendel's process, Mendel's seminal work was accomplished using the garden pea, that is Pisum sativum, to study inheritance. The flower petals remain sealed tightly until pollination is completed to prevent the pollination of other plants. The result is highly inbred or true breeding pea plants. These are plants that always produce offspring that look like their parent. By experimenting with true breeding pea plants, Mendel avoided the appearance of unexpected traits in offspring that might occur if the plants were not true breeding. The garden pea also grows to maturity within one season, meaning that several generations could be evaluated over a relativity of short time. Finally, large quantities of garden peas could be cultivated simultaneously, allowing Mendel to conclude that his results did not come about simply by chance. When we see the Mendal's law of inheritance, they are fundamental principles of genetics, which is discovered by Gregor Mendel in the mid-19th century through his work with pea plants. His experiments laid the foundation for the field of genetics. His laws describe how traits are inherited from one generation to the next. There are three main laws. Number one is law of segregation. Number two, law of independent assortment. Number three, law of dominance. Mendel conducted his experiments using pea plants which had easily observable traits such as flower color, seed shape, and pod color. He cross-pollinated plants with different traits and observed the inheritance patterns over several generations. His meticulous record keeping and statistical analysis led to the formulation of his laws. And also, Mendel postulated that genes, that is characters, are inherited as pairs of alleles, which is called as traits, that behave in a dominant and recessive pattern. Alleles segregate into gametes such that each gamete is equally likely to receive either one of the two alleles present in the diploid individual. Inheritance can be defined as the process of how a child receives genetic information from the parent. The whole process of heredity is dependent upon inheritance, and it is the reason that the offsprings are similar to their parents. This simply means that due to inheritance, the members of the same family possess similar characters. Between 1856 to 1863, Mendel conducted the hybridization experiments on the garden peas. During that period, he chose some distinct characteristics of the peas and conducted some cross-pollination, that is artificial pollinations on the pea lines that showed stable trait inheritance and underwent continuous self-pollination. Such pea lines are called true breeding pea lines. This is a diagram shows the different shapes of the seed, colors of the flower, and also different structures of the pods, and also the combination of different structures of stems. When we see the law of dominance, here when we see the key points, that dominant alleles mask receives alleles in heterozygotes, and also recessive traits are expressed only when two copies of the recessive alleles are present. Mendel discovered that by crossing true breeding white flowers and true breeding purple flower plants, the result was a hybrid offspring. Rather than being a mix of the two colors, the offspring was purple flowered. He then conceived the idea of heredity units, which is called as factors, one of which is the recessive characteristics and the other is dominant. Here the diagram shows that the cross between the violet, that is purple color, and white. Here we are getting the results in the F1 generations all with the purple colors. Then they are self-fertilized, and during the F2 generations we are getting the ratio of 3:1. That is 3 numbers of purple and 1 white color. In that ratio we are getting the results. Plants which are used in first generation crosses were called P or parental generation. Plants, Mendel collected the seeds produced by the pea plants that resulted from each cross and grew them the following season. These offsprings were called as F1 generations. Once Mendel examined the characteristics in the F1 generation of plants, he allowed them to self-fertilize naturally. He then collected and grew the seeds from the F1 plants to produce the F2 or second filial generation. Mendel's experiments extended beyond the F2 generation to the F3 generation, then F4 generation, and so on. But it was the ratio of characteristics in the P, F1 and F2 generations that were the most intriguing and became the basis of Mendel's postulates. Upon compiling his results for many thousands of plants, Mendel concluded that the characteristics could be divided into expressed and latent traits. He called these dominant and recessive traits respectively. Dominant traits are those that are inherited unchanged in hybridization, and also the recessive traits become latent or disappear in the offspring or a hybridization. The recessive trait does, however, reappear in the progeny of the hybrid offspring. An example of a dominant trait is the violet colored flower trait. For the same characteristics, white color flowers are the recessive trait. The fact that the recessive trait reappeared in the F2 generation meant that the traits remained separate in the plants of the F1 generation. Mendel proposed that this was because the plants proceed to copies of the trait for the plant color characteristics, and that each parent transmitted one or their two copies to their offspring where they came together. Moreover, the physical observation of a dominant trait could mean that the genetic composition of the organism included two dominant versions of the characteristics, or that it included one dominant and one recessive version. Conversely, the observation of a recessive trait meant that the organism lacked any dominant versions of these characteristics. Second one is about the law of segregation. Here, this law states that each individual has two alleles for each gene, one inherited from each parent. These alleles segregate or separate during the formation of gametes, that is, egg or sperm. That each gamete carries only one allele for each gene. When gametes unite together at the time of fertilization, the offspring inherit one allele from each parent, restoring the pairs of alleles. When we see the key points, number one is about each organism has two alleles per gene. The second one is allele separate during gamete formation. Third one is each gamete carries only one allele for each gene. Then the fourth one is fertilization restores the pairs of alleles. This law states that the alleles of different genes assort independently of one another during gamete formation. This means the inheritance of an allele for one gene does not affect the inheritance of an allele for another gene, provided the genes are on different chromosomes or for apart on the same chromosome. According to this law of segregation, only one of the two gene copies present in an organism is distributed to each gamete, that is, to the egg or sperm that it makes, and the allocation of the gene copies in random. When an egg and a sperm join in fertilization, they form a new organism whose genotype consists of the alleles contained in the gametes. Let us see something about the test cross. A test cross is a cross between a person with an unknown genotype and another person with an homozygous recessive genotype. A test cross determines or discloses the genotype of the original person. A test cross can assist in determining if a dominant phenotype is homozygous or heterozygous for a certain trait. This cross is a genetic technique discovered by Gregor Mendel that entails mating an individual with all phenotypically recessive individuals to ascertain the zygosity of the former by evaluating the proportions of offspring phenotypes. Zygosity can be homozygous or heterozygous. Heterozygous individuals have one dominant and one recessive allele. Individuals with homozygous dominant alleles have two dominant alleles, whereas individuals with homozygous recessive alleles have two recessive alleles. The alleles inherited from the parents define the genotype of an offspring for each of its genes. The alleles combination is the product of the maternal and paternal chromosomes provided by each gamete during the fertilization of that offspring. During meiosis in gametes, homologous chromosomes undergo genetic recombinations and randomly separate into haploid daughter cells, each with a unique combination of paternally coded genes, and also dominant alleles will overpower recessive allele expression. The genotype ratios may be derived using mathematical probability, but the genotype composition cannot be determined merely by looking at the phenotype of a dominant feature. It is impossible to determine whether a tall plant from F1 or F2 generation has capital T or capital T and small t combination. As a result, Mendel crossed a tall plant from F2 with a dwarf plant to identify the genotype of a tall plant at F2. It is known as a test cross. Instead that of self-pollination, a dominant phenotypic organism is mated with a recessive parent in a normal test cross. The offspring of such a hybrid is simply analyzed to predict the genotype of the test organisms. Then Mendel also came up with a way to figure out whether an organism with the dominant phenotype was a heterozygote or homozygote. This technique is otherwise called as test cross and is still used by plant and animal breeders today. Let's assume that you were handling a tall pea plant with no knowledge of its parentage. Since tallness is a dominant feature in peas, your plant may be homozygous or heterozygous, but you have no idea. In this case, what will happen? That this cross can be used to establish its genotype. If the plant were homozygous, a test cross would produce all tall progeny. If the plants were heterozygous, the test cross would produce half tall progeny and half short progeny. Any recessive allele in the unknown genotype is expressed in the progeny of a test cross because it is linked with the recessive allele from the homozygous recessive plant. The crossing of an F1 progeny with the double recessive parental progeny is done in order to determine whether the progeny is homozygous or heterozygous for a character under consideration. This test cross is performed to determine the genotype of a dominant parent if it is a heterozygous or homozygous dominant. On the basis of the results obtained, the ratio of the offspring, it can be predicted that the parent is having which genotype. If all the offsprings are of the same phenotype, then the parent is homozygous dominant. If there is 1:1 ratio in the phenotype of the organisms, the parent is heterozygous dominant. This diagram shows the possibilities of the test cross. Then when we see the monohybrid cross here, monohybrid cross is a cross between two individuals who have homozygous genotype for the opposing phenotype for a certain genetic characteristics. Here the genetics utilize monohybrid crosses to study how homozygous children express heterozygous genes acquired from their parents. In this experiment, Mendel took two pea plants of opposite traits and crossed them. He found the first generation of offsprings were tall and called F1 progeny. Then he crossed F1 progeny and obtained both tall and short plants in the ratio of 3:1. To know more about this experiment, we can see the monohybrid cross in detail. A dihybrid cross is a cross between two distinct genes whose observable features differ. It is a result of a cross between two heterozygous people for two different separate features. Let us see something about the law of independent assortment. Here, when we see the key points here, genes on different chromosomes are sorted independently. Then the inheritance of one trait generally does not influence the inheritance of another. And also this law states that when an individual has two different alleles for a gene, one allele can mask the expression of the other one. The dominant allele trait is expressed in the phenotype, while the recessive allele trait is not unless the individual has two copies of the recessive allele. Mendal's law of independent assortment states that genes do not influence each other with regard to the sorting of the alleles into gametes. Every possible combinations of alleles for every gene is equally likely to occur. In independent assortment of genes can be illustrated by the dihybrid cross, a cross between two true breeding plants that express different traits for two characteristics. When we see here again in the dihybrid cross, here the genes were observable features and also they differ with each other. It is the result of cross between two heterozygous people for two separate features. Consider the characteristics of seed color and seed texture of two pea plants. One that has green, the another one is wrinkled seeds, and another with yellow round seeds. Because each parent is homozygous, the law of segregation indicates that the gametes for the green wrinkled plant all are capital Y and small r, while the gametes for the yellow round plants are all capital Y and capital R. Therefore, the F1 generation of offspring all are capital Y, capital R and small r. This is a diagram explains the dihybrid cross. When we see why the pea plants were selected by the Mendal's experiments, there are so many reasons behind that. He selected a pea plant for his experiments because of they are easily grown and maintained very easily. They are naturally self-pollinating plants but can also be cross-pollinated. It is an annual plant. Therefore, many generations can be studied within a short period of time. And also, it has several contrasting characters. Then Mendel conducted two main experiments to determine the law of inheritance. The experiments which includes monohybrid and dihybrid cross. And also, while we do experimenting, he found that certain factors were always being transferred down to the offsprings in a stable way. Those factors are called as genes. That genes can be called the units of the inheritance. When we see the significance of Mendal's laws of inheritance here, his work went largely unnoticed during his lifetime, but was rediscovered in the early 20th century. His principles provided the basis of the modern genetics, explaining how traits are inherited and paving the way for the discovery of DNA as a genetic material. Understanding as the Mendel laws is crucial for studying genetics, breeding programs, and various applications in medicine and agriculture. They also provide insights into the mechanisms of evolution and the diversity of life forms. When we see the results, main results from the Mendel experiments, the genetic makeup of the plant is known as the genotype. On the contrary, the physical appearance of the plant is called as phenotype. The genes are transferred from parents to the offspring in pairs known as alleles. Then during gametogenesis, when the chromosomes are half and there is a 50% chance of one of the two alleles to fuse with the allele of the gamete of the other parent. And also when the alleles are the same, they are known as homozygous alleles. When the alleles are different, they are called as heterozygous alleles. When we conclude his experimental evidences, here working with green pea plants, Mendel found that crosses between parents that differed for one trait produced F1 offspring that all expressed in one parent traits. The traits that were visible in the F1 generation are referred to as dominant, and the traits that disappear in the F1 generations are described as recessive. When the F1 plants in Mendel experiments were self-crossed, the F2 offsprings exhibited the dominant trait of recessive trait in the 3:1 ratio, confirming that the recessive trait had been transmitted faithfully from the original parent. Reciprocal crosses generated identical F1 and F2 offspring ratios. By examining sample sizes, Mendel showed that traits were inherited as independent events. Thank you.